Hello, madam.
Hello. Yeah,
Yeah,
good
very good.
morning.
Good morning, ma'am.
Good morning.
Madam, the section going to be started at 9:30, ma'am. Is it is
Yeah,
fine
yeah.
for you?
I know. I'm
Yeah.
fine. So I'm
Okay,
really
ma'am.
only just to check whether I'm able to log in or not. So that
Okay,
is the
ma'am.
reason I have logged in. Otherwise, we'll
Okay, ma'am.
continue at 9:30. Yeah.
Yeah, ma'am.
Huh. Okay.
Okay, ma'am.
Hello.
Prabhakar madam, good morning, madam.
Good morning.
How are you, madam?
Fine.
Okay, thank you, madam.
Madam, shall I start the section?
Okay, ma'am.
Prabha ma'am, are you there?
Yes.
So shall I start the section?
Yes.
Ashwini madam, carry on.
Okay. A very good morning to all. Myself, J. Ashwini, Assistant Professor, Department of Pharmacology, acting as a chairperson for this AICT and IICT sponsored refresher program on role of artificial intelligence in drug discovery and development held during 25th January 2022, 1st February 2022. All of us gain a lot of knowledge from previous session and I hope the same further too. Now I hand over the session to coordinator of the program Dr. A. Lakshmana Rao Garu Principal VVIT Institute of Pharmaceutical Sciences, Gudlavalle, to speak of few words.
Thank you, Ashwini Garu. A very good morning to all the participants and respected speaker of the today's session, Dr. Prabhagar Madam, for the day 6 of 1 week online AICTE ISTE refresher program on role of artificial intelligence in drug discovery and development, organized by VV Institute of Pharmaceutical Sciences, Gudlavalledu, Krishna District, Andhra Pradesh. Today, totally, we are having 4 sessions, 3 plenary lectures and valedictory function. And session 1 is taken over by Dr. Prabhagar, Associate Professor, National Institute of Pharmaceutical Education and Research, Naipar, Mohali, Punjab. She will speak on prediction of protein function using machine learning approach. And the chairperson for this session is Ms. J. Ashwini, Assistant Professor. Next, session 2 will take over by Dr. Girinath G. Pillai, Research Scientist, Nairo Research India, Bionest, Kochi, Kerala. He will speak on role of statistics and machine learning in lead optimization using ADMAT. And the chairperson for the second session is Ms. M. Chandana, Assistant Professor. Next, session 3 will be taken over by myself, Dr. A. Lakshmandra, Professor and Principal, VV Institute of Pharmaceutical Sciences, Gudlavalledu, AP. And I will speak on role of artificial intelligence in drug discovery and development. And the chairperson for this session is Ms. Wasimunnisa, Assistant Professor. And finally, the session 4 is valedictory function. And the chairperson for the valedictory function is Ms. A. Saidatri, Assistant Professor. Now, I hand over the session to Ms. J. Ashwini Garu, chairperson for today's first session, to introduce the resource person to the participants. Over to Ashwini Garu.
Ashwini ma'am, turn on your video and audio.
Okay. It's my privilege to introduce the speaker of this session, Professor Prabhagar. Professor Prabhagar is working as a professor and the head of the Department of Pharmacoinformatics in Naipar, SAS Nagar. She did her BE and MTech from IIT Rourke and PhD from Tapur University in Patilla. She works in the area of artificial intelligence, database and development, data analyticals. She did publish more than 65 research papers in peer-reviewed journals, developed approximately 20 software and databases, delivered 15 invited lecturers, guided 5 PhD and 60 master students. With the brief profile, I welcome you, sir, to talk. Thank you, sir, for sharing your knowledge with me. Thank you for.
Thank you for the kind introduction. So myself Prabhagar. So I will be sharing first my PPT. Is it visible?
Yes, madam, it is visible.
Prabhagar ma'am, before sharing your PPT, can you switch on your video first?
Switch on the video.
Yeah.
That thing, no,
Yeah.
madam, is visible.
Yes, ma'am.
Can I switch off the video, because otherwise sometimes the network issue, the voice doesn't go properly because of the network bandwidth. So
No, ma'am. I just want to take a picture of you. That's why I asked you to switch on the video.
So to continue with the prediction of protein function using machine learning approaches, all the participants are attending the various lectures. They have heard about the various applications and the other thing. Where I'll be talking about the prediction of the protein function, like how we have applied the machine learning approaches in the predictions, as well as I will show you the web server which we have developed. Before that, I would also like to give you the basic steps which are very important whenever we are working with the AI applications or the machine learning approaches to be implemented for any one of the area. The steps are very generic, like they are not specific to the protein function, but they can be applied to any other area. So before that, let us try to find out what is the intelligence when we are talking about on the artificial intelligence. Normally, when we are talking about the intelligence, we are talking about the data, which is comprised of the basic information or the basic values which are being used. For example, this is the data which I'm showing is for some gene expression data. The values are the gene expression values for some platform, and these are the expression values. If you see them individually, they do not give you any type of the information. So this is the data which is generally unfiltered, and it does not give you the, but this is the does not give you any information, but these are the actual values which you are collecting from the various platform. When you process this data, for example, if you have taken the microarray data or the gene expression data, you can always find out which are the genes being expressed by doing the analysis on this or by doing the processing of this data. That gives the information. You have received the information after processing this that these genes are being expressed, whether they are the down-regulated or the up-regulated genes. But still you do not have whether they are really contributing for the, or whether you can use them as a biomarker or not. So after that, the knowledge resides in the users, which is applied to the application, and that is like you are extracting the knowledge from this data. And the intelligence is the application and integration of the knowledge. So the artificial intelligence is basically the capability of a device to perform the functions that are normally associated with the human intelligence. Like the people who are working in the biology or the bioinformatics, they can, or the people who are working on this gene data, they can, by seeing the various values, they may be having some kind of the information. They can just do the analysis that which genes are maybe contributing, which may not be contributing, based on the availability of the information or the data. They are involved in the various pathways. So the capability of a device to perform functions that are normally associated with the human intelligence. Now, if this type of the information or the intelligence is being performed by the machines, that is known as the artificial intelligence, because you are training a machine which can perform the intelligent behavior, or which can work as an intelligent machine, which is doing some intelligent work. So they are the intelligent machine, and we say it is a artificial intelligence. AI is the study of how to make computers do things at which at the moment people are better. You are not creating any AI based applications on which the people are not having the expertise or something. So you need the people which are able to perform the work, or they can do the prediction, or they can do the analysis of the data. AI is the science that automates intelligent behavior. It is a system that thinks and acts like humans and the rationally. So basically, when you are working with the artificial intelligence application, if you are having a problem, to solve those problems, to define the rational behaviors, or the ability to act like humans. For example, the robotics applications. You develop the robots which are acting like the humans. So these include learning, means when you build the model, you train the model with the existing data. And from this data, the AI algorithms extract the rules for a specific applications. So whenever you want to use this AI-based model, these rules are applied to the new data for the predictions of the data. For example, the weather forecasting, or maybe when we are talking about the protein function, whether we are trying to find out the enzyme class, or maybe the subcell plot, or maybe the any other type, or maybe the structure of the proteins, whether it is a tertiary or any other type of the structure, whether it is a alpha helix or the beta sheet, whatever be the type of the structure we are trying to predict. Initially, we train the model with the existing information. Then you have the new protein or the new data based on which you are going to do the prediction. And based on this prediction, some of the algorithms are also there which does the self correction, because if it is not predicting correctly, so it does the self correction, so that in future you are having the better predictions. So the machine learning is an approach in achieving the AI, or the intelligent behavior, that utilizes the computers and mathematical algorithms to build the models based on available data sets. When we talk about the word machine learning, it is not a single algorithm. You work with the number of algorithms, and depending upon the type of the algorithm or type of the application, you select the appropriate algorithm. For example, you can use the SVM, or the support vector machine, where you are using it for the classification. For example, the example of the subcell predictions. What you do is, if any one of the protein is given, you can find out its subcellular location. So it is a classification problem where you are associating the input data with a specific class. All of you are appearing for the exams. The classification is being done based on the grades, grade A, grade B, grade C, or maybe they are classifying based on the pass or the fail, or maybe you are being evaluated based on the marks. That is the out of 100, say you are getting the 80, 90, 50, 60. So these are the different type of the application, and based on that, the application algorithms are selected, whether it is the support vector machine, the KNN, the PNN, or the artificial neural network, or the fuzzy logic. So not a single algorithm, there are the hundreds of the algorithm which comes under the machine learning algorithm. And broadly, what we can categorize these algorithms in the supervised, unsupervised, and the reinforcement learning. When we are talking about the supervised learning algorithm, at the time of training or building the model, with the input value, you are also having the output value. If this is the input data, this has to be the output value. Y is equal to fx type of the applications you are trying to fit in this supervised learning algorithms. So input and the output both are provided to build the model. While in this unsupervised learning, you do not have the output variable while you are building the model. Then how it is going to predict? There is a basic difference. When we use a supervised learning, basically they are used for the regression type of the applications and the classification algorithms. In the unsupervised learning algorithms, we try to use it for the clustering. Clustering is, you'd say this is the similar type of the input data. You do not say whether it will behave like this A or like B. You say the input data is similar to the this previous data. So you just define, and what we say in the protein function, if the sequence similarity of the two proteins is very high, we say they may be behaving similarly. So you can predict the function based on the similarity of the input sequence. In the reinforcement learning, when you give the data, there is the output. This output is used to correct or train the model. That is the reinforcement learning. Mostly it is used for the games or the robotics, or maybe the driverless cars also. These are some of the steps which we have to follow while we are working with the machine learning process. First, you have to define the problem. None of the machine learning model is generalized normally. None of the model in today's time, also when we are using extensively this machine learning or the artificial intelligence, there is no general algorithm. You always define the problem and the scope of the problem for which your model will be built. Then you have to define the identification of the required data. Identification of the required data, for example, if you try to predict the points within this square, like on your PPT, and at the building of the model, you provide only the data for one corner. And if you try to predict something on this corner, this is say your problem domain, then it may not predict it correctly. So the identification of the required data which represents the problem domain is very, very important. If your data is not correctly identified, maybe the model, whatever be the good algorithm you are applying, that may not predict correctly. For example, all of you might be seeing the opinion polls or the exit polls at the time of this elections. So when you see these opinion polls are failing, or whether they are doing the correct prediction based on the data itself. If your data selection is not correct and the algorithms are not correct for the selection of the data, it may give it. Then the data pre-processing is the next step. Then the defining of the training set and the test set. Now the algorithm selection. Here we talk about which machine learning algorithm we are going to use. Whether it is genetic algorithm, fuzzy logic, ANN, SVM, KNN, PNN, so many algorithms are there. So based on the problem, you may have one algorithm. And normally when you are building the model, sometimes you do not use a single algorithm. You try to build the model based on more than one algorithm and see which algorithm is performing better. Then you use this algorithm based on this training data set, you train the model. Then you evaluate this data model based on the test data. Here you are defining, you have the total data, you divide into the training and the test data. The built model on this training data, you evaluate with the test data. If the test data is being predicted correctly, then you say the model is built. You can use it for the classification or the regression analysis or anything. In case it is not predicting correctly, again, maybe you are trying to tune the parameters. Any algorithm you take, there are the number of variables which you have to tune with algorithm. For example, in the SVM algorithm, you have to define the C and the gamma value, which are not fixed. These values are again optimized based on the data. So the C and gamma values are the variable. If you use the ANN, how many hidden neurons and what are the number of layers. So there are always the variables which have to be defined by the users for the same algorithm. So you may have to tune the parameter. Maybe you have to use a different algorithm. Maybe you have to divide the data into the training and test set. Maybe somewhere the data pre-processing or the identification. So if your data model is not predicting correctly, all the steps you have to see carefully again. So it is a iterative method. Number of times you will apply the algorithm to finally build the model. And when we are talking about, because nowadays the word data science, data analytics are very well used. So data science is the extraction of actionable knowledge directly from the data through a process of discovery, hypothesis, and an analytical hypothesis. You use in this the statistics, data mining algorithms, you use the mathematical algorithms, you need the programming skills, you need the analytics system, you need the domain expertise and the research. The combination of all the things is the data science. So in the data science modeling, as I told you, the algorithm is based on the type of the problem. You may be having a problem like the scoring, predicting or estimating a numerical value such as price or a probability. When you get the marks out of 100, that is a type of the algorithm we use for the scoring algorithms. For example, if you try to find out the IC50 value of the molecules, all the molecules against a specific protein, if you try to find out whether they are active or inactive, say for the Alzheimer or any other disease, you try to predict their IC50 value. And this IC50 value is giving a numeric value, which may range from some 0 to say 3, 4 or some nanomolar or the micromolar. So this is a type of the scoring algorithm. Ranking algorithm is learning to order the items by the preference. For example, you are trying to predict the proteins which may be for a specific disease. You are saying that these proteins are involving. So if you get the 10 proteins which may be like the target for a specific disease, you want to prioritize them. For which protein you want to work. So the ranking, Like when you are giving a rank in the class, first, second, third, fourth, or in any one of the competition, you get the merit list. The similarly for the proteins which are identified for a specific disease or the therapeutic category, you would like to order them in the preference, which is more important or which may not be the more important. So the clustering is the another type of the algorithm where you are grouping the items into the most similar groups. Classification, deciding if something belongs to one category or another. Finding the relation, finding correlation or potential causes of effects seen in the data. Characterization, very general plotting, and the report generation from the data. So these are some type of the model building tasks or the modeling tasks which may be performed using this machine learning algorithms. In the data analytics, when we are talking about, there are basically the four types of the applications which we work upon: descriptive, diagnostic, predictive, prescriptive. Descriptive means what is happening. So at the time of COVID, you could get that the people are having this type of the how many people are suffering from this COVID and what are their symptoms. So you are just observing and recording what is happening. You do not analyze why it is happening. So diagnostic type of the data analytics is when you try to find out the reason. Why is it happening? The reason why the people are getting the COVID, then it comes the problem in the diagnostics. So this is the type of thing. Predictive, when you try to predict the value for something. For example, if you try to predict the function of a protein, basically that comes in the predictive category. Prescriptive is what do I need to do? For example, if you want to stop the COVID, what it should be done to stop this? If you can build a model that is known as a prescriptive, basically this type of the models are very rarely you can say more or less not existing as on date. Very few models which are of the type prescriptive, not in the case of the drug discovery, but maybe in some of the applications. But these are very, very highly intelligent models which are comes under the category of prescriptive modeling. So when we are talking about the data pre-processing, so that is the data handling part. So what are the examples of the input data is very important. Now in the input data, what characteristics are distinguish the class examples? For example, when you are defining the data, always you talk about the variables x1, x2, x3. For example, if you are trying to predict the function of the protein, how the protein will be predicted for a specific function. If you give the input sequence, it will not directly predict the protein function. This protein sequence you have to convert into the characteristics or the features or the variables, whatever the term you want to use. So this protein sequence is converted into some numbers or some categories based on which the data will be built. So which features are the variables which may predict the function of the protein is very, very important. You can generate thousands of the descriptors. Say you can count the amino acid composition, you can have the dipeptide composition or the tripeptide, but exactly which features are the important is the selection. If you have the unimportant features at the time of model building, that may mislead you. So the selection of the features is again important. Again, you have the various algorithms, maybe the Baruta algorithm or the recursive feature elimination or the backward elimination algorithm. So you have the number of algorithms for the selection of the features which are important for the problem you are working. So how will discriminatory evidence be combined to make a decision? Means you are trying to identify the algorithm which is performing best on your data. Then finally, you have to do the assessment with how well your model is working. So you use the cross validation, you use the leave one out or the other methods for the assessment of the model. In the data preprocessing also, you use the various steps like in the data cleaning. Data is cleaned through processes such as filling in the missing value. When you collect or generate the data for some of the input data, some values are missing. whatever the reason, maybe the software you have used to generate the data from the protein sequence to the descriptor, it is not able to generate for some of the protein some value. For example, when we use the small molecules for the generation of the features, and if these molecules are having some of the matters, some of the softwares are not predicting the features for these molecules. So you have some of the missing values. And if you are collecting the data from the various sources, for some of the molecules, you may not be having the data, or maybe the for the protein. So how to handle this type of the problem? Either you remove the complete row or maybe the feature. And this decision is not without analyzing the data. You have to analyze the data, whether you have to remove the data point or the features for this. So there are the various ways, or maybe sometimes you fill these values by some mean or the median value of the same class. Then the data integration, when you collect the data from the various sources, there may be some conflicting evidences. For example, if you are collecting the protein data or the sequences or maybe their functions, when you are collect the data, you will find out some of the inconsistency or the conflicts. You have to analyze the data. You have to remove these conflicts from the data. Data transformation, that is the normalization, aggregation, and the generalization. I'll give you the example. For example, if you are simply you are taking for the protein the amino acid composition. So if you are having a protein which is having say that 100 amino acid or say the 1000 amino acids in a protein sequence. From this 1000 sequence, there are the 20 amino acids. For each amino acid, you will count how many amino acids in this 1000. So for this 20 amino acid, the total has to be 1000, but it may happen the first amino acid is having only the 5 number, and the another amino acid is having the 120 or the 30 or maybe the 300. So there is a lots of difference in the values of the number of amino acids for the 2 amino acid. You try to convert into 0 to 1 or maybe say 0 to 10 or any range between minimum to the maximum, so that the data is within a given specified range. That is known as the data transformation. Data reduction is, this steps aims to represent a reduced representation of the data in a data warehouse. You always divide the data into the training, test, or sometimes maybe the validation data set. You do not take the whole data for the model building. You have to test the model based on the test data. So you do the data reduction. Data discretization involves a reduction of a number of values of a continuous attribute. For example, if you are collecting a patient data, whatever be the clinical research or any other data, so the patient age is a continuous value. So, or maybe the weight of the people, you do not use exactly the weight. Normally, if you say the age is divided into maybe say 0 to 14, 14 to 28 or 30, and then. So that is known as a data discretization. Depending upon the features, you may use this value also. In the data cleaning, for the filling of the missing values, there are the methods that you ignore the value use. So these are the various methods. I'm not going into the details of this, but you have to apply one of the methods. When you are doing the data cleaning or from the data, you will find out some of the outliers are there. For some of the data points, you see that the one or the two values are not as going in the same, like similar to the other data points. They are very, very different. They are known as the outliers. I will show you in one of the application, not for the protein function, but I will show you for the blood-brain barrier permeability application, how we have identified the outliers. So every time you identify the outliers, now you have to analyze it is exactly the outlier, means due to some collection of the data or due to some noise in the data, the outliers have been inserted, or it is some novelty in the data which is defining some new thing in the data, the new information in the data. For example, these outliers in the fraud detection in the banks or the credit or debit card are very well used, or in the computer security. So sometimes they give you the information, or sometimes you remove them because they are misrepresentation of the data. So again, you have to analyze the data at the time of cleaning with this. These are some of the methods for the normalization, mean max, or the decimal scaling, or the Z-score method for the normalization of the data, any one of this. When we are talking about the data reduction, reducing the number of attributes, you are doing the feature selection, means the number of columns you are reducing, or the attributes or the features or the variables. Any term, these are interchangeably used with the different applications. So you are reducing the number of attributes, you are reducing the number of attribute values when you are doing the data discretization. The continuous values, maybe you are just converting into some specific range of the value. Then you may be working with the reducing the number of the tuples or the number of the samples. You do not use the whole data for the model building. For the variable or the feature selection, there are the various types of the algorithms. We use the filter methods. These are, they apply the statistical measure to assign a scoring to each feature. And in this filter methods, like this is the forward selection. What it does, in the forward selection, you define the p-value or maybe p-value or the significance of the each feature has to be less than the p-value. Then you create a simple regression model. And for each feature, you will find out in the data set what is the p-value. And any one of the descriptor which is having a high value, you keep on removing that. And again, you will build the model. So that is a forward selection, how you are selecting the models, or the backward selection when you are removing the features from the whole data set. So these are some of the methods which we use for the feature selection. For example, in the Baruta algorithm, when we are working with the feature selection, what it is done, you are having the 10 descriptors or the 10 variables. You may generate the 10 shadow features, which will be having some random values. And when you build the model for each features, you will try to find out whether it uses the random forest algorithm and it identifies the importance of the each features in the random forest algorithm. And in the random forest algorithm, when the feature's importance is measured, if the shadow feature is having a higher importance than the actual feature, it is removed, means it is not important. And finally, you get the important features in this. For the data validation, you are working with the training and the test set. You are dividing the data set. The next algorithm is you work with the holdout method. This is training set, test set. You use this data set for the model building and evaluate with this test data set. And most of the algorithm, when you are working with it, at each step, you use the evaluation of this. Number of epoch or the iterations are used. For example, in the artificial neural network, you will stop the algorithm as soon as it reaches a point where the error in the test set is minimum. You stop this. Or the validation set, it is stopped, then you stop the training of this. Then you may be using the k-fold cross validation. What is done in the k-fold cross validation? You divide your input data into the k parts. K can be 5, k maybe 10, depending upon the size of your data set. For example, if you are using a 5-fold cross validation, you divide the data into the 5 parts. The 4 parts are used for the training of the model, and the 5th part is used for the evaluation of the model. Then you will include this 5th into the training set and remove another one part for the evaluation. That way 5 times you will build the model and each data point is being evaluated. And finally, you see how well it is performing by using the mean square error, or in the regression by using the R square value. And whenever you are getting the best value that you define, this is the best model. Leave one out, rather than you are dividing into the 5 or the 10 parts. Every time you remove one data point and build the model, and then you evaluate this left out data point for the model validation. Then once you say that your model has been built, now you have the new data set. Whether this new data set or the new point can be predicted or not, you have to check the applicability domain. In any one of the machine learning model, it is always having the applicability domain. It does the extrapolation up to a limit, but if you are having a totally very different input data which is not having any type of the similarity from the training data set, it may not be predicted correctly. It may be by chance predicting correctly, but you cannot rely the data prediction. So the applicability domain is always evaluated. So in the applicability domain, is a test compound similar to the training data set or not? That is being checked. For example, this is the applicability domain. This is one variable. This is another variable. There are the algorithm. You draw the box plot based on the various values. Now you say the external data points are very similar to the input data points. This is the range of the values. They are very similar. But if you see some of the data points somewhere at this point, say the right bottom corner, none of the input data point is here in this area. So the prediction for this input data may not be reliable. That is how you check the applicability domain for the new data set before you apply the model. So this is the descriptor 1, descriptor 2. This is the range of the descriptor 1 and descriptor 2 for the training model. So whenever you are working with the new data points, that should be somewhere near to this range. It can go somewhere slightly outer than this box, but not very different from this input data. Now let me talk about some of the application which we have used for the prediction of the protein function. I'll give you more two examples for the protein function. One is for the subcellular localization prediction. Another is the AC number prediction. AC, the enzyme class number, that is the 4 levels, and then these 6 types of the protein functions which have been defined, and the up to the 4 levels it goes. For example, in this we have used the data used for the trainings collected from the UniProt from this release database. The following filters we have employed for our application. The organism field was used as the eukaryotes. Proteins which were localized in more than one subcellular compartment were also removed. Initially, we used a single subcellular localization. Later on, we have used a multiple subcellular localization also. And proteins which are having less than 30 amino acid composition, because the feature generation was creating a problem, and having any ambiguous amino acid composition, amino acids, for example, BX, this were removed from the data set. Finally, from this data sets, we were having the 38,354 protein sequences. Now, to produce a non-redundant data set, number of proteins are very similar to each other, and if we use this whole data set as it is, it may create a biased model where the number of proteins are similar in the data sets. So we use a CD hit program, and none of the protein is having more than 90% of the identity, and this is the threshold value we have used. So any protein which is similar to the existing protein in the training data sets were removed from this. Now for the test set, again, to remove the biasness from this removed points, we have just applied again the CD hit, and none of the protein is having more than 25% of the sequence identity, so that our test set is not biased for a specific class. Now for finding out the features from these protein sequences, we have used the primary protein sequence. We have not used the secondary or the tertiary structure, simply use the protein sequences for generation of the features or the variables or the characteristics. So the first 20 features are based on the amino acid composition. So as I have given you the example, if you are having the 1000 amino acid in a sequence, you count for each amino acid, how many amino acids are there in this sequence, and then we have normalized by using this formula. F1 is the number of numbers for that specific amino acid. For a given protein, which amino acid is having the minimum value? The maximum minus minimum. So we have normalized the data so that the variation in the numbers is removed. So the 20 features we have used from the amino acid composition, and the remaining 9 features, because it says 29 dimensional vector, means we are using the 29 features. The 9 features we have used based on the amino acid order or there is a sequence order, as well as we have used some of the properties of the amino acid. In this, we have used the hydrophobicity, hydrophilicity, as well as the mass of the protein. For the amino acid indexes, we have used from the online sources. And this is the equation which we have used. In this equation, we have taken the amino acid sequence order up to the 9th. So we take the first amino acid, we take the 3 properties, mass, hydrophobicity, and the hydrophilicity for that given amino acid, and we see the next amino acid. Again, we take these 3 properties and we use this equation. And this we have used up to the 9th order. So the 9 features we have used based on these equations. So the total number of features we have used is the 29 for this specific application for the model building. And basically this we have used based on our previous, not our previous, based on the previous work published on by Dr. Chaw. So we have taken this application and created a 29 features. In this, we have used the 2 algorithms because it is a classification model. So we have used the classification algorithm KNN and the or the K nearest neighbor and the probabilistic neural network. And the metric function which we have used is the cosine and the Euclidean distance. Why we have used this 2 algorithm is, I don't know whether the students are very well aware of this KNN and the PNN algorithm or not. In the K nearest neighbor, what it does, when you take the new protein sequence, it will identify based on these 29 features. It will find out if you are having a 29 features for the new protein and all the training data set, you are having the 29 features for each data point. Now it will calculate the angle between these 2 vectors. You can draw them as the vectors. It will calculate the cosine or the angle, because if you see, if you create the 2 vectors, any one of the vector may have the same angle but the different Euclidean distance, and with the same Euclidean distance you may have the different angle also. So with the K nearest neighbor, we are trying to find out the localized or the nearest proteins behavior we are using. In the probabilistic neural network for the prediction, we are using the whole data set. These are the 2 very different algorithms which is taking the effect of the nearest proteins, and the another algorithm is taking the considering all the proteins in the training data set. And the 2 different type of the metric functions have been used, the cosine and the Euclidean distance, which work on a very different. Now this is the amino acid indexes which were used for the hydrophilicity, hydrophobicity, and the mass, which we have downloaded from the internet for this amino acid, and we have used them. This is the data which we have not generated, we have just downloaded from online and used this data in our application. So in the classification model, we do not use the R square value or the correlation coefficient. We use the precision, sensitivity, specificity, accuracy, and the Matthews correlation. And these are the formulas which are used. True positive in this applications are, if a class is predicted correctly, a true class means a positive class is predicted positive, that is known as a true positive. False positive means if it is negative class and it is predicted positive, then it is a false positive. Similarly, the true negative and the false negative. So based on the predictions, we find out the value of TP, TN, FP, F1 and calculate the accuracy, sensitivity and the precision. These 5 parameters we have calculated. So when we have used the model for the validation, we have used the 5-fold cross validation for the KNN as well as for the PNN method, which are used. Initially, we have used this 11 and the number of subcellular localization. And if you see, the number of sequences are varied. The data is not balanced. It is imbalanced. Still, we are able to predict the classes up to a very good, like the predictions are very good for this algorithm. Similarly, for the test set, if you see the specificity very much near to 1, very much near to, except the vacuole where only the 29 sequences were there. Here it is less. For the 27, it is less, but the specificity is very, very good for this. So even if the data is very much imbalanced, we are able to predict the values for this. Similarly, for the PNN methods, we have used the 5-fold cross validation. 5-fold cross validation means the whole data was divided into the 5 subsets, and 1 subset is used for the test, and the 4 subsets were used for the model building. Next time, this subset is included in the training set, and from the training set, 1 subset is removed for the test set. So these are the values, the precision values, the sensitivity, specificity, which were very good. Similarly, for the test set, we have calculated this. Now, how we have predicted, because we have used the 2 algorithms. Now, for the final predictions, what we have done, we have used for the KNN what is the angle between the 2 proteins, means the nearest protein and the new protein which we want to predict. And if the angle is between 0 to 6, the prediction accuracy we identify in the 5-fold cross validation is this, and in the test set is this. While for if the angle increases, the accuracy is keep on decreasing. So which is very much validating our information that as the angle between the 2 proteins means they are very much dissimilar. And this information, this is what we say in the data analysis we did, that if the angle is between the 2 protein is this, then the accuracy in the 5-fold is this and the test set is this, like this. This information was used for the final prediction. So if in the KNN method, it predicts the subcellular localization A, and for the PNN, it does for the also the subcellular localization A. These are not the amino acid. Just to give the examples, A is the name of the subcellular and this is the subcellular, and the angle is between this. So we say the probability between 0 to 1 is 0.85. Both the algorithms are predicting, which are very different in nature, the same subcellular location, and the angle is also very less. So we say the probability of the correct prediction is 85%. If both are predicting the same, but the angle is more than this, we have reduced the probability of correct predictions because the angle has increased. But if both the proteins are predicting the different algorithm, then and the angle is less, still we have reduced because the 2 algorithms, one is based on the nearest proteins and one is based on the total data set. Again, we have reduced the probability because the 2 algorithms are not predicting correctly. This is the way we have given the probabilities of the prediction, because when it is a classification algorithm, we have taken, say, the 11 subcellular location initially. It will predict one of the subcellular, but the new protein may not belong to any one of the subcellular. It may also happen. So if that may be the case, the angle will be more. And if the angle is more, the probability may be very, very less for this. So this is how we have built the model for the This is the screen. Let me share the prediction screen. How we have used this. Are you able to see the screen for the predictions? The web server page? So this is the prediction. Here we used to give the protein sequences and then it gives the predictions. For example, if this is the screen where we type the protein sequence, and if we select the KNN algorithm and say the predict, this is how it says. This is the EC number for the given, and this is in the application we have entered the values. If this is the EC number is being predicted, this is the name of the class, and the probability of this specific prediction by using the KNN algorithm is this. And if we are using the PNN algorithm by giving the probability is 0.95, this is the same class which have been predicted, and we can identify this. And in case we change the protein sequence and again we use on this, it will ask for this. It will just take 1 or 2 minutes and doing the predictions. Now you have changed. Now the remaining sequence is more similar to this, but the probability has been reduced. It is not similar to the previous. So how the predictions have been done for the enzyme class. Similarly, we can use for the subcellular localization also. If I just say this is the protein sequence, and if we say submit query, it will does the 1 minute. So this is the same type of the screen. When we paste the protein sequence here and say submit, we get the name of the protein sequence. Initially, what we developed in the protein function is for a single subcell. After that, we added the data into the application for the multiple subcellular localization also. And we were able to predict the subcellular localization. So the same algorithm, same criteria was used for the EC number prediction, what the application I have shown you. And this was the, not this one. Yes, this is the example for the subcellular localization that we have extended it for the EC number prediction. KNN and the PNN algorithm, again the cosine and this, and the number of classes are more than 4,000 in this data set, not 1 or 2 classes. Because the EC number, if you say like 5.3.1, so up to the first level, second level, third level, fourth level, total number of classes are around 4,000, which it was able to predict very, very much reliably, we can say. I would also like to give you one more example, though it is not for the protein function, it is for the blood-brain barrier permeability. Just to give you the example for the outlier analysis. In this, what we did, it is the small molecules, whether we want to find out whether they cross the BBB blood-brain barrier, or the log BB value is lower or the higher. Because for the diseases like Alzheimer or the Parkinson, the molecules must reach to the brain. While in the other type of the therapeutic application, they should not cross this blood-brain barrier pattern. So in this, we have collected the data of 191 molecules, and the features which we have collected for this is the molecular weight based on the previous knowledge published on the research papers, that topological polar surface area, C log P, PGP substrate probability. The descriptors were used for this model building. When we built the model for this, we divided the data set into the training and the test set. Initially, 141 molecules were in the training set and the 50 molecules were in the test set. After building the model, the artificial neural network was used, and this is the architecture which we have used: 7521. Means 7 is the input layer, 5 is the first 5.
Prabhagar ma'am, madam, madam, are you there? Madam, Prabha ma'am, are you there? There might be a network issue from the Prabha ma'am side. All the participants, please wait a while. Sorry for the inconvenience, all the participants. There is a network issue from the madam side. Please wait for a while.
Hello.
Yeah, ma'am. You are audible now.
Okay. 1 minute. Just 1 minute. Let me share my.
Sure, ma'am.
Is it visible?
Yes, ma'am. It is visible.
So in this prediction of the log BB value of the blood-brain barrier permeability, we have collected the molecules. We have used the supervised function approximation algorithm, which is the artificial neural network. The architecture which we have used, maybe the students are not aware of the ANN, I don't know. So, but this is the architecture which we have used, and this is the log sigmoid, and the linear transfer function has been used. So these are the parameters which we have defined, and we have used the MATLAB software. So when we have built the model, we identified there are the 9 molecules which are not being predicted correctly, and they are the outliers we have identified. Why we tell them that they are the outliers? These 9 molecules were there, and most of the molecules from this are from the neuromuscular blockers, and experimental log BB value of all of them were extremely low. Like these molecules are very, very different. If the value for the remaining molecules, say we are having say between -5 to say +5, they were having -25 or something like this. This is the value of the log PV. So they are very different. And when we have analyzed their data, some of the molecules, their molecular weight is very high. That is the reason that maybe the log PV value is very low. For some of the molecules, their C log P value is very low, or the PGP substrate value is very high. So this is the reason, as I told you, when you are working with this, you simply do not remove the molecules which are not being predicted. You have to analyze them. So after this analysis, we remove these 9 molecules and build the model. And after building the model, we receive the very good prediction values for the correlation coefficient, coefficient of determination, and every parameter is very nicely predicted, and that is for the training set as well as for the test set. Working set means we have calculated for the total data set of the molecules. After this, we try to compare it with the already known commercial softwares or the free. So this is the Series 2 softwares which we have used, CSBB, which is freely available, and the pre-ADME software. These are the 3 softwares with which we have compared our model. And for the given test set, this model is giving the R value 0.66, while our model is giving 0.90, as the R square is 0.44 and 0.81. So from all these 3, our model was having the much better predictability in the one. There is one more application which we have developed, that is to say the drug target interaction. In this, we have used the protein data as well as we have used the small molecule data for the model building. So that is known as the proteochemometric approach. And for this proteochemometric approach, we have used the data set from the various sources in this. The maximum time we face the problem with this proteochemometric or the PCM model is the non-availability of this non-interacting data set. We have the information that this small molecule is interacting with this protein, but we do not have the data for the non-interacting data set. And when we are trying to find out the prediction whether a molecule is targeting a specific protein or not, we need both the positive as well as the negative data set. Otherwise, we cannot build the part. So we have used a rational method for selecting the negative data set. So what we did first, we have identified which is known for the interacting pairs. Then for the non-interacting pairs, we have used the chemical, this Tanimoto similarity coefficients. So the ligand should be similar to the positive data set. based on their chemical dynamic and based on the physicochemical Euclidean distance. But the distance between the positive data set and the negative data set must be very, very high. So this is a strategy we have used for selecting the negative data set, or the decoys data set was generated for building the model, which was very critical for this. So because it is a PCM approach, we need the data for the drug as well as for the target, and the bioactivity data sets from these various sources have been used, like the drug bank, Campbell, PubChem, PDSP, Stitch, Binding DP, and the therapeutic targets databases. We try to use the primary as well as the structure information, 3D structure information, and build the model. Finally, in the after building the models with the 3D structure as well as from the primary protein sequencing, we identified there is not much difference between the 3D information to be used. So we preferred to use the primary sequence information. So when we collected the data from this source, we have used this SCPDB data source. In this, the ligand protein or the drug target complexes are 9877. Distinct proteins are 3000 in this, and different ligands are 5339 in this. So ligands were inspected and prepared in Discovery Studio. This is the software which we have used for generating their structure. Binding sites were annotated to the UniProt accession numbers, and the complete protein sequences were retrieved from this UniProt database. Then we did the cluster-wise pairing, like the clustering has been done so that any pair which is not, or any cluster which is having the less than 10 entries were removed from this data set. Because it is a very large problem domain, we do not want to have the imbalanced data, so we remove this data. And after doing the analysis with respect to the organism, how the data was distributed, this is the distribution of the data based on the enzyme class, the first level, the 6 classes: hydrolase, lysis, isomerases, ligases. These are the 6 classes how the data was distributed. And based on the structure, because as I told you, we use a 3D structure, how the resolution values were. This is the distribution of the data. And based on the organs, like the Homo sapiens data is this much, from the others is this much, like E. coli and all these are the sources of the data for this. Now for the external set was collected from the Campbell drug bank and the therapeutic target data set. And in this we have extracted the IC50, PIC50, KI, PKI. Based on that, we have defined the positive and the negative data set. And finally, in this, we were having the 91,000 non-redundant interactions covering 593 targets and these many new ligands. Then we have built the model. For the descriptor calculation, we have used the Prophet server. We have used for the structure-based, like these are the 2 web servers we have used for the protein descriptors, and Morgan fingerprints were calculated using this RDKit software for the small molecules. For the model development, we have used the SVM, RF, and the KNN algorithm as well as the NetBase. These are the variables, like the number of trees was kept as 100 for the RF classifier, value of K, 1, 3, and 5 were kept for the KNN algorithm. For the SVM, C, and gamma were also optimized. So after doing this SVM model, RF model, these are the values. ROC value, recall, specificity, precision, accuracy. These are the values which were calculated from the various models which we have built. After predicting the, building the model for this drug target predictions, all the predicted targets for the nandrolone, this is the drug which we have taken, belongs to the Homo sapiens and have been reported to play an important role in cancer. So we have taken this molecule and we have predicted the targets for this. These are the 5 targets which were predicted for this. And the APP score and the predictability score was very good for this. And when then we have docked them, docking score was also good for these 5. So we predicted the targets on which this drug may be acting for this nedolon phenylpropionate. Similarly, we have taken this anthracyclines against the with aldose reductase 1B, and this is the APP score. And these are the generic names of the drugs which were identified to be working with this much of the drugs. So this is the information how we are doing the prediction. There is one more thing. For example, we have used this anti-cancer drug, sorafenib, in the prediction model. For this, these 7 targets were known, already known. But for these, we have identified 3 more targets which this may be acting. And based on this drug targets, we can identify their pathways, how they are working, and we can find out the more information. Not only for a given drug, which are the targets, we have taken a target which may be the drugs working from the data set on which they are doing. And we have also defined their APP as well as the docking score. For example, for this drug, everything we have taken, I think this is from the natural sources we have taken, and this is also from the natural sources. And these are the targets on which this drug is working. 1, 2, 3, 4. These 3 are from the Homo sapiens, which it has identified, and this is the Trypanosoma cruzi. This is the target from the. This is the organism for this target which we have identified. They are working. So if we have any target which are very much similar to this, maybe from the Homo sapiens, it may also work. So this is how we have used the various types of the applications for the prediction of the, or maybe for, we can say, for the use of the machine learning approaches. So it is not only for the protein function, you can try to find out the IC50 value the way we have predicted the log BB value. You can also find out whether a given molecule is interacting with a given target or not. So you can define any type of the problem, and you can use the ANN or the AI or the machine learning methods for this. With this, I'm just finishing my lecture. So any question on that? Any question?
Okay. Dhatri madam, okay. Shall I start, madam?
Yeah, you can.
Participants are requested to unmute yourself and ask the questions. If you have any doubts in the today's topic, you can unmute yourself and ask the questions.
Thank you, madam, for sharing your knowledge with us. Dear
Thank you.
participants,
Yes.
I request the participant to share your queries, if any. Feel free to ask, or you can post your queries on chat box. Ashwini madam, conclude the section.
okay. Okay. Thank you, madam, for sharing your knowledge with us. On behalf of VV Institute of Pharmaceutical Sciences, it's my honor to present on e-certificate. Please accept it, madam.
Okay, thank you. Thank you.
Thank you very much, madam.
Thank you.
Saidati garu.
Yeah, sir.
Next session will start at 11:30. Now
Sir,
it
it
is
is
10:45.
11:15.
Okay. Oh yeah, it is 11:15 only. Ah, you
Yeah.
can start at 11 o'clock if possible.
Sir is already at a busy schedule, I thought. So
Okay, I
Pillai
think.
sir is only available at 11:15.
Okay. Okay, sure. You start at sharp 11:15. Huh? Okay,
Yes, sir.
okay.
The feedback and
Ashwini,
assessment.
Okay.
One
Ashwini
second.
garu, you announce it
Okay. I
for next
request to
session.
all the participants fill the feedback link, feedback and assignment. The link will be provided in the chat box. The next section will be started at sharp 11:15 a.m. Please join again and thank you all.